one of the learning outcomes for the session on nerve tissue is to explain nerve transmission, and that's what we're going to aim to do in this video here. From the reading that you've already done and the worksheet that I've asked you to complete before this, you should be quite familiar with the neuron as a cell in itself, and so we can see from this diagram here that's what we're looking at. We've got a cell body with the dendrites that extend from that, and that's kind of like the head, the top of the cell. And then we've got the length of the axon, and at the end the terminal boutons. So depending on where the cell is and what its function is, this cell could be microscopic in length, or, for example, if it's within the brain, it might be traveling a very, very small space over a very small space. If, however, it was, say, the sciatic nerve, which runs from the base of the spine right down the leg, that neuron could be a huge length, and it's the axon, the long, thin part of the nerve, that travels that distance. We describe a neuron as being irritable and as being conductive. So what we mean by irritable is that it's able to generate an impulse, and conductive means that that impulse is able to travel the full length, which could potentially, as we've just said, be meters. So the nerve impulse will always travel from the dendrites of the cell body to the terminal boutons. It will never travel in the other direction. Therefore, we have nerves that go from our central nervous system, so from our brain and our spinal cord, to our extremities. So the cell body will be in the central nervous system, and the terminal boutons will be at wherever they're actioning. But we also have neurons that travel from the extremities back to the central nervous system, because it's a two-way process. The brain needs to tell your hand what to do, but your hand needs to tell your brain what, for example, it's feeling. So if you're touching something hot, if you're experiencing pain, we need to get that back. And because the impulse will only travel one way from the dendrites to the terminal boutons, we would need two nerves, one that goes there and one that comes back. And we'll talk about that a lot more when we look at the actual nervous system. But for today, we're really just focusing on the neuron itself, and in particular just now, how the impulses move along the length of that. And that happens through a process called action potential. So the definition that I've put there is obviously from a dictionary, so it's a little bit wordy, but we're going to explain that. So just to read it through then, action potential is described as the rapid change in electric potential that parts of a nerve cell undergo when a nerve impulse is generated. So leaving it at that for a moment, we are talking about an electrical impulse. It's quite specific to nerve cells and to some extent muscle cells as well. However, it is an electric impulse. Now, normally when we think about current, for those of you who have done physics in your previous learning, when we think about a normal flow of current, we're talking about a flow of electrons. But what creates current in action potential is movement of ions from outside the cell into the cell and back again. And it's about sodium and potassium ions. It needs to be initiated by something. And that will either be initiated by the cell itself, because it's irritable, so our neurons within the central nervous system, or our neurons at the periphery, our sensory neurons, will detect something and generate that impulse. Or the impulse could come from another neuron, and there are times when one neuron meets another neuron and the impulse passes from the terminal boutons of one to the dendrites of another, and then it would be conducting the impulse along the length of it. But it's all about this exchange of sodium and potassium ions across the cell membrane of the neuron, in particular across the cell membrane that surrounds the axon of the neuron. And because some ions are positively charged and some ions are negatively charged, the charge of the cell changes. So, this diagram probably looks a little bit complicated, but when we talk through it, it will make a lot more sense of the things. If we focus on this aspect for now, so these 4 pictures here, or diagrams, what we're looking at here is the plasma membrane. So we'll go right back when we talked about the membrane structure. You've got your phospholipids that have got their heads on the outside and their tails on the inside, and they're end to end with each other. And then we have proteins that insert into that membrane. In this case, integral proteins, they go from one side right to the other, and they control the passage in and out of the cell. Now, in a normal resting state, when the neuron is not being activated, it's not being stimulated, sorry. If we look at this green, in the first diagram, this green protein molecule, it's controlling passage of things in and out of the cell that the cell needs. It needs nutrients, it needs oxygen, that type of thing. However, it is not allowing the sodium and potassium ion to change place. So in its resting state, intracellularly, we have these orange dots, and those are potassium ions, and they have a negative charge associated with them. So the internal aspect of the cell is negatively charged. I'm just going to highlight that so you can see. So negatively charged internal aspect because of these potassium ions. The external environment of the cell, however, in the tissues, in the fluid, the extracellular fluid, that's full of sodium ions, and they're positively charged. So, positively charged exterior surface because of the sodium ions that are outside. The proteins which control the passage of the potassium and the sodium at resting state are closed. The potassium can't get out and the sodium can't get in. So, negatively charged cell in a positively surrounded environment. However, when the nerve is stimulated, that all changes. The protein that controls the passage of general substances, the oxygen and the nutrients and stuff, that closes over. So nothing can use that pathway. And all of a sudden, the protein that controls the sodium ions opens. Now, a cell will always want to try and be neutral with its surrounding environment. So, as soon as this channel, this gate, opens, the sodium ions flood into the cell because the cell's not very pleased about the fact that it's very positive on the inside and very negative on the outside. It's trying to find a neutral position. So the sodium ions become flooding in, and that changes the polarization of the cell. It goes from being this negative environment on the inside to a positive environment on the inside. And that is where your action potential is generated. It's referred to as depolarization, a change from a negative to a positive environment because the sodium gate has opened. And what we can see from the graph here is that the cell rests normally at this level, but all of a sudden, as the sodium ions flood into the cell, it becomes positively charged and a current is generated. Now that will only happen for the period of time in which the cell is stimulated, which is a very brief moment. As soon as that stimulation stops, this sodium gate closes again. That's what we've got in this third diagram. It's closed over. And the cell pumps those sodium ions back out. Because, again, we're trying to neutralise with the surrounding environment. We were at the stage where we had a positive on the inside and a negative on the outside because the sodium ions had flooded in. So now we're going to get them all back out again. So the sodium gate closes and the potassium gate opens. And the potassium goes back out. And the cell returns to its normal state, where it is positively charged on the outside and negatively charged on the inside. And then the cell goes back to its resting state, where the normal channels for normal nutrition can open as, and the cell can proceed in its resting state. So we were up here because the cell had been depolarized, and now as the sodium leaves the cell and the potassium remains, the current will drop and it will go back to its resting state. And this happens all along the axon of the neuron. It's not the case that the entire cell, the potassium gate opens and then closes. It happens almost like a Mexican wave all along the length of the cell. So it does happen at some point the whole way along, but not all at the same time. And that's what we can see from this diagram here. The impulse is generated from the dendrites, and we've got positive and negative changing along the length like a Mexican wave. So depolarization and repolarization are occurring at the same time along the length of the cell. And depending on how far that is, that will obviously dictate how long that process takes to happen. Always from the dendrites of the cell body along the axon to the terminal boutons. So, excuse me for the sort of double background thing going on here. This is a slide from a previous presentation that I've stolen. Just to sort of summarise, when the cell is resting, we have extracellular sodium, which is positively charged. Inside the cell, we have potassium ions, which are negatively charged, and the membrane is closed. The potassium can't leave and the sodium can't enter. However, when the nerve is stimulated, the membrane permeability is altered. The sodium ions flood into the cell and depolarize, make the cell positively charged. Once the stimulation has stopped, the sodium ions leave the cell again and repolarization occurs and the action potential stops. So, from your reading, you'll have found out that some cells have got this fatty sheath around them called myelin. In fact, most cells, most neurons, are myelinated. You do have some unmyelinated, but in general, most neurons are myelinated. And the myelin is there to provide insulation and protection for the length of the neuron. However, it means that the cell membrane can only be permeable in the areas where there isn't the myelin sheath, because otherwise you have the extracellular environment and the intracellular environment are separated not just by the membrane but also by this myelin sheath. So along the length of the axon, the neuron has, the myelin sheath has little notches, the nodes of Ranvier. And those nodes are really important because that's where this action potential can take place. And because it can only take place at spots along the length of the axon, it makes the process quicker. Because rather than, let's go back to this diagram here, it having to happen the full length, it can jump bits. It can happen here, and then we can go to here, and then we can go to here, and then we can go to here, because the myelin sheath is protecting the rest of the way along there. So it makes the transmission of the impulse much quicker. It's as if the action potential is jumping from one to the other, and it can all happen at a much faster rate. So myelin isn't just about protection and nourishment, it's about speeding up the transmission as well. But it can only happen in the areas where the myelin sheath isn't there, at the nodes around the egg. So that's what happens when the impulse goes from one end of the neuron to another. But we just need to think a little bit about what happens to that impulse when it gets to the end of the neuron. Like I've said before, sometimes you have neuron meets neuron, so the impulse passes from one cell to another. And other times you have the neuron meets the tissue that it's affecting. And for the purpose of this, we're going to say it's muscle, because if we're thinking about a neuron that goes from the central nervous system out to the tissues, it's going to be affecting the muscle because it's giving us a reaction. So when we have two neurons that come together, that's called a synapse. When we have a neuron meeting muscle tissue, we call that a motor end plate. And that's what we're looking at here in sort of very simple diagrammatic form. So if we think this is the terminal bouton of one neuron, and this is the dendrite of the next neuron, the impulse cannot pass through action potential because this is one cell to another. There is a gap between them. Similarly, when you have neuron meeting muscle fibre, there has to be a way for the impulse to get from the terminal boutons into the muscle and affect the muscle. And this happens through a process of chemical nerve transmission. So the impulse gets to the terminal boutons, and the cell releases a chemical that then passes through the synapse, so through the gap between the two cells, and the dendrites of the next neuron absorb that chemical, and that kicks off the action potential process in the next cell. And these chemicals are called neurotransmitters. The chemicals are made by the endoplasmic reticulum that you find in the terminal boutons of the neuron. So they produce this neurotransmitter, which is packaged by the Golgi apparatus, sent to the membrane, and then released through the membrane, where it then passes through, using the protein channels, passes through the membrane of the next cell and into there. Now, neurotransmitters, there's lots of them, and they're chemicals that you may have heard of. It's things like dopamine, serotonin, acetylcholine. Sometimes we call them happy drugs, happy hormones. Now, they're not hormones, they're neurotransmitters. But often we think of particularly serotonin as being a happy thing. It's when it's released in our brain, it makes us feel happiness and pleasure. And it's being released by neurons because it's passing impulse from neuron to neuron. So synapse between neurons and motor end plate if it's the neuron meeting up with the muscle that it needs to affect. So that's what's really covered, how impulses pass from along the length of an axon. It's a really important thing to get your head around, particularly in terms of the relationship between the sodium and the potassium ions. And one of the reasons why it's so important is when you come to the beginning of second year, when we teach you how to give local anaesthetic to a patient. Local anaesthetic works by blocking the proteins that allow the sodium ions to enter the cell, and therefore the cell cannot repolarize and the impulse cannot pass along that cell. So it's important that you understand the sodium-potassium process, otherwise how local anaesthetic works will not be clear to you. And obviously if we're going to give local anaesthetic to people, we need to understand why it's working. So that is one of the reasons why understanding nerve impulses is important. It also helps when you go on to talk about human disease and we think about times when, because of disease, nerve impulses don't pass. Diseases like MS or multiple sclerosis is one of those things. So as always, go ahead and do a bit more reading if you need to, and if there's anything you're not clear on, please bring it with you to the tutorial and we'll discuss it at that point. Thank you. Bye-bye.